I bring you thanks from the advisory panel, part of the group that reviewed, that set the challenge became Mutographs, reviewed the panels that went through. And I spent just a few minutes telling you a little bit about the process and a little bit about the view on Mutographs now as it goes on. So the first thing I would say from the advisory panel is that really have three major jobs, two of which you'll recognize if you've been involved in major reviews in the past, pretty common operations. The first thing, obviously, was reviewing the applications as they came through. Pretty standard, but they're quite large grants, but that's the only thing that was a little different. And then several of us have been involved in the yearly reviews or the visits to come and see you, like today's operation, watch the progress that goes on. Again, pretty standard. The one thing that's a little different was actually setting the challenge. So the panel itself is charged with every couple of years coming together and setting the grand challenges. And so we've had about 19 of them so far that have gone out and have funded now a series of 7 different teams. And I might say a little bit about that process, what a grand challenge is to us. It's a little hard to define, I have to say. Obviously, this is an experiment. Nobody really knows how well this is going to work, although the progress you all are making, I think, sort of makes you feel pretty confident that the operation is behaving well. We're obviously looking to fund team science, international, and very large in the operation. And in putting together the grand challenges, we tried to be very specific and thinking a little bit about what a grand challenge should be, as opposed to just being a large project grant that needed to be funded. And so the panel spent a fair amount of time discussing what kind of characteristics that we wanted the grand challenges to be. And the first thing that we wanted them to be was to be a goal that was outside of our reach, that was not dramatically far away, but something that wasn't immediately obvious that you could go to. It was going to need a special framework to get there, and we were hoping that we could nucleate special interactions that could really drive those populations, that kind of possibilities. And the thing that was really key in setting these things, it was the sort of thing that you realized when you heard about it and defined it. It was something that you realized that if success happened, it would be a big impact on our field as it went through. And another thing I want to say, particularly since many of the CRUK people have gone, I can say they took a big chance in doing this. Still are, I think, in some ways. They've invested a lot of money in this process in a sort of international collaboration that one wasn't really sure about how it's going to go. And so I think they deserve an awful lot of respect for being willing to take that challenge. And a second thing in their agreement in setting these up, which was bold on their part, is they told us as the committee, the teams don't have to succeed. So I do a fair amount of work for the National Cancer Institute, and I can tell you NCI would not put this kind of money into programs that they thought had a chance of not succeeding. But CRUK put their money on the line and sort of said, we want you to be out there far enough that this is people working at the edge of doing things and seeing that to go. And so I think they deserve a kind of an interesting little plus in your minds about going through this. Since I think most of them are gone, they hopefully won't be embarrassed by me saying that. I thought I also might tell you, kind of pull back the curtain a little bit about our discussions about mutational signatures in the committee as we were thinking about whether we would actually set a grand challenge in this area. Because it was a stepwise process, and as our vision for it grew, we got more enthusiastic, and I'll tell you a little bit about that step. So we all enjoyed reading about the original papers about the identification of new carcinogens and how this opportunity of using mutational signatures could be used to generate that kind of information. But I have to say, at that stage, we sort of thought this community, you all, would do that anyway. We weren't really sure that a big project, just identifying mutational signatures and linking them to other new carcinogens, was something that was going to fit the grand challenge opportunity. We liked it. We liked talking about it. But as our discussions went on, began to take on other features that we thought might be worthy of the grand challenge. The first thing, which we thought this might be very disruptive to epidemiology. And I mean that both in a positive and negative way, about how epidemiology might be changed dramatically by being able to sequence DNA and figure out what kinds of exposures had happened, even rates of those. And I have to say, our interest rose considerably when that happened. We also thought it was a starting point for new biochemistry, trying to understand how carcinogenesis happened with new agents. That was an important thing. And what pushed us over the line was the understanding that this could be a big event in prevention. And when we sort of began to realize how you could put these things together in prevention, it became a package for us that we thought mutational signatures would make a terrific grand challenge. And so we began crafting the language that worked out for the original challenge. You all obviously applied and succeeded. And you brought all sorts of new things in that we hadn't even understood could be possible. And so, you know, the whole idea about how you could look into normal tissue and understand the importance of carcinogenic events in normal tissues that went on wasn't clear to us when we started out. It wasn't clear to us about how we could understand how deeply the animals and cell line work was going to contribute. Nor did we really understand how this might actually become precursors and diagnostic for different therapeutic approaches, which I think is still a little bit of a possibility as opposed to a really strong opportunity. But those were all areas that we as a group didn't understand. But I think you guys have done an unbelievable job. You're about halfway through your 5 years of funding that you've gone through here. I have to say, personally, I've enjoyed every single moment I've come in contact with the team, both in the yearly reviews and these little check-ins that go up every 6 months. At each moment that I've come, there's been some display, some new set of data that's changed my mind about what things were important and weren't important. As a scientist, that's something you love. That's just something that just thrills everybody when you get that moment where you kind of go, oh, I didn't get that. And it's, you know, it's been Ludmil, and it's been Peter, and it's been Alan, and it's everybody kind of uncovering new flavors of this problem and what you can do. And that's been just absolutely terrific as we've watched that go on. The team interactions are great, and I get the possibility there may be the opportunities to interact between the teams, which again was something we didn't imagine at the beginning part, but that's good. So there are now 7 teams, so there's 7 rooms like this across the world of people who sort of are into this large-scale interactive science, and I think that's a really important aspect. And so I just want to say thanks. I mean, I've heard great science. I've met good people. It's been a real joy for me to come in and join you all in watching this happen. Obviously, this is not my field. I get to just watch. A bit of a voyeur, I think, in this setting, but a very enjoyable position to be in. And that's what I wanted to say. I can't imagine there are any questions for me, but I would be happy to answer any as we go through. But I want you to know that the panel and CRUK, everybody is very pleased with the development of the Mutographs project. Good job. Don't stop. Go hard. Okay? Thank you.